parallel of X64 and XRF. But in order to study the function of parks and the genetic interaction, for example, between parks and XRF, first I have to introduce what is genetic interaction. A genetic interaction is defined as a deviation from the expected phenotype when combining multiple mutations. For example, an organism has both gene and A and B, they are viable. But if one of the genes is mutated, then the organism is still viable. But if both of them are mutated, then the organism is lethal. So in this case, we can say that gene A and B, they have genetic interaction. Or to be specific, you can say that they are synthetically lethal. And this comes to this morning's trial lecture, the PARP inhibitors used for treatment of BRCA1 or BRCA2 mutated cancers. That is to use the synthetic lethality between PARP and BRCA1 or BRCA2. So we generated the PARP knockout mice in our lab and found that the PARP knockout mouse, they behave very similar to wild type, so there's no overt phenotype. And also in 2008, XRF knockout mouse was reported to have no overt phenotype compared to wild type. However, we found that when we combine PARPs and XRF together, double knockout, the mouse is lethal. So we proposed a possible model to explain the lethality of the double knockout. That is, we think that PARPs and XRF, because they have similar structure, so maybe they can compensate for each other. For example, when PARP was deficient, then XRF can compensate the role of PARPs, so the double-strand break can still be repaired. And vice versa. So when XRF is deficient, then PARPs can compensate the role of XRF, and the double-strand break can still be repaired. However, if you have both deficient, then the double-strand break cannot be repaired efficiently. Then the accumulating breaks can lead into cell death. Maybe that's why the mouse, they are temporarily lethal. So to verify our model, we proposed another protein, P53, which is known as a tumor suppressor. Also, it can prevent genome mutation. But how? So P53 can help activate DNA repair proteins when it senses DNA damage. And also it can arrest the cell cycle so that the DNA repair protein can have enough time to repair the breaks. However, if there are too many breaks, then P53 can have to initiate the apoptosis process, then the cells they die. So we found that we asked if this lethality between PARPs and XRF double knockout mice is dependent on P53. And we found the answer is yes, because when we inactivate P53, the triple knockout mouse, they are viable. However, the triple knockout mouse, they are significantly smaller than their siblings, and this is a quantification of the mouse weight at day 30. And also, we want to test if the immune system is affected or not. So we isolated the thymus and found that in the triple knockout mice, the thymus also significantly smaller compared to the siblings. And also by splenocyte counting, so this is a statistic. And in addition, we analyzed the spleen. We found that the spleen size also smaller compared to their siblings, and also by splenocyte counting. So with this, we conclude that the PARPs and XRF, they have genetic interaction, and the double knockout mouse, they die embryonically, and this lethality is dependent on P53. So you might wonder, are there any other factors that can compensate the role of XRF except PARPs? And to answer this question, I have to introduce another factor, DNPCS. So DNPCS is short for DNA-dependent protein kinase catalytic subunit, and it has 4,128 amino acids, which means it's very huge. And the protein size is around 450 kilodalton, So it's a hitch-based kinase in humans. And the DMPTCS, it can interact with the Q70-Q80 heterodimer, and they can function as a huge complex. So we want to study the genetic interaction between XRF and DMPTCS. And in 2013, my supervisor, Mengtan PhD, he found that the double knockout mice of DMPTCS and XRF, they also die, although the single knockout, they are viable. So we asked the same question, whether this double knockout DMPTCS XRF mice also depend on p53. And we found the answer is yes, because when we inactivate p53, even after insufficiency, can rescue this quality of DMPTCS and XRF mice. However, the triple knockout mouse, they are also significantly smaller compared to their siblings by their look or by quantification. And in addition, we performed another assay, which is telomere fluorescence in situ hybridization assay, and it's short for T-fish. So for this assay, we have to collect the mouse tail fibroblast. We cut the tail and then we collect the cells, and we put the cells in culture. This is how they look like in microscope. And then we add cosmate. Cosmate can inhibit the microtubule growth, and then it can arrest the cell cycle in metaphase. So afterwards, we collect the metaphases and fix the metaphases on the slice, and we use the pepsin treatment to digest the proteins around the chromosome so that the chromosome DNA can be exposed. And then we hybridize with DNA probe, which can complementary with the telomere ends. Afterwards, we stain with therapy, and then we took images. So here is an example of the mouse metaphases, how it looks like. And when you zoom in, you can see like the intact chromosome has 4 red signals, which are the 4 telomeres. And here shows the example of chromatin break, where this one red signal is far away from the other 3. And here shows the example of chromosome break, so there's only 2 red signals.